All right, we can go ahead and get started. Welcome everybody to week 3 of our DART MS for siege drug analysis course. This week we'll be hearing from Amber McConnell and Jennifer Benetti on the state level perspective of using DART mass spec in their casework, as well as using high resolution mass spectrometry in their analyses.
All right, so Jennifer's and I are gonna switch off and on, so I'm gonna ask her to advance slides for us. But we're both gonna talk about what it's like to bring DART TOF into a state laboratory system. So you can advance to the next slide. So a disclaimer, the opinions expressed here are our own and do not necessarily reflect those of the Virginia Department of Forensic Science or the Maryland State Police. Next slide. Basically, this presentation is going to go over why we chose to implement DART at our laboratory, what you might need to think about if you're not sure if you want to bring DART into your laboratory, how we validated the instrument, some choices we made along the way, how we're using the instrument, so whether or not we're using it for screening or confirmation, and just some final considerations along with some tips on how to testify to the technology. Next slide. So if you're like me, I started in the field 18 years ago, and the field has changed a whole lot since I started. When I started, there was about half the samples were marijuana, and it took about 5 minutes to analyze them. We saw a lot of cocaine, some heroin, and on an exciting day, we would get an ecstasy tablet. That's no longer the case. We see a lot of novel psychoactive substances. They're in all different types of forms, like liquids, coated on paper, edibles, counterfeit pharmaceuticals. So it's no longer as easy to use color tests to screen these samples to get an idea of what's in there. So we applied for an NIJ grant several years ago, and we were given the money to purchase the DART TOF, as well as we were going to develop targeted methods for 3 different classes of drugs. So it would be cathinones, synthetic cannabinoids, and fentanyls. Another reason why we wanted to consider implementing DART is because we were starting to see some dangerous fentanyls in the lab. They were starting to get more and more potent, and we wouldn't realize what we had on our bench until we already had the GCMS results and we had already weighed it and handled it. So we wanted to see if we could screen for these substances without having to open the packaging in case we were potentially exposed to carfentanil or one of the really potent fentanyls. And finally, the idea was also that maybe we wouldn't have to have a GCMS method that could screen for everything, that we could screen using the DART and know whether or not this substance was actually negative for CDS and maybe stop at DART. Next slide. So the DART is actually a rather large instrument. It's about the size of a washing machine, and there's a lot of things that need to go with it. So it takes up a lot of space in the laboratory. We didn't have too much of a problem finding a space in Pikesville because we had a lot of open area in Pikesville, but you do want to make sure that it's not super close to where you're handling a lot of different solvents, because that could affect your analysis. And we also didn't have a 230-volt line in the middle of the laboratory. So we had to have an installation of the 230-volt line. And they did recommend that we had a UPS. So the next slide will show you all of the stuff that also is near the DART. So you have the large tanks that have the helium and nitrogen. And we got a tank holder bolted to the floor. You also, so the big box on the left in front of the DART is the UPS, and the line coming down is the electrical line. And then you have the pumps for the DART, and the bottom right picture shows the portable snorkel. We recommend a portable snorkel because when you're analyzing with DART, you're actually volatilizing the sample in atmosphere, so you don't want to be exposed to any of the fumes. Next slide.
Okay, so now I'm going to start talking about how we validate the method for casework. So our central lab got our first DART in 2007, which I believe is probably one of the first forensic labs to begin to investigate the DART for forensic drug screening. If I'm incorrect, someone can feel free to correct me, but as far as I'm aware, that was kind of one of the earliest. Because of that, Bob Steiner and Robin Larson actually published our method validation in the Journal of Forensic Science. So if you're interested on kind of more details than what I'm going to be providing here, you can feel free to look up that publication. But in general, they investigated 3 different separate topics, including a lower limit of detection study, selectivity, and then also they looked into how the reproducibility was compared to established techniques. So I'll go into each of those 3 different experiments that they did. So they first looked at the lower limit of detection. And of course, there's no way to determine this for literally every drug that they might come across when screening samples. So they just sort of did a representative sampling of 7 different drug compounds from various classes. So they looked at alprazolam, butalbital, cocaine, heroin, methamphetamine, testosterone, and trazodone. And what they did is they made solutions of these 7 drugs, which they acquired via purchasing primary standards, and they were prepared at 5 different concentrations in methanol. The acceptance criterion that they set was that the spectra at the orifice 1 voltage of 20 volts had to show the measured mass of the protonated molecular ion at plus or minus 5 millidaltons of the calculated mass. And then they determined the overall lower limit of detection to be the dilution level just above where that acceptance criterion failed. Once they had determined that, they then did 10 subsequent analyses of each drug performed at that limit of detection just to demonstrate a repeatability that if we're saying this is our limit of detection, we can actually detect these substances repeatably and reliably at that level. And so they noticed that the acceptance criterion began to fail at 0.03 milligrams per milliliter for 2 of the drugs. So they determined that 0.05 milligrams per milliliter was approximately a lower limit of detection that worked consistently for the drug classes that were studied. Now, if you're used to validations for maybe more toxicological purposes, that might seem like a sort of very loose definition of a limit of detection. So obviously, again, we were looking at this for screening, so this wasn't kind of meant to be a be-all, end-all, this is our absolute limit. It was really just to get a sort of more general idea of the sensitivity of the technique. And I also want to point out that this was an older generation instrument, and so when the acceptance criterion was beginning to fail, it wasn't that the drugs were not being detected, but that they were detected outside of that point, of that 5 millidalton range. And so nowadays, our instrument in my laboratory is now a 4th generation instrument, which has a higher resolution and increased precision. So it's possible with the newer instruments that lower limit of detection could potentially be decreased and have higher sensitivity. In addition, they also did this with function switching, which obviously, as you've heard, has a lot of benefits, but it can potentially sacrifice sensitivity. So if you're very, very interested in really increasing the sensitivity, you could just focus on having only 1 voltage be analyzed. For the selectivity study, they looked at 5 different pairs of drugs that had the same molecular formula to determine whether they could be differentiated. So they looked at meth versus phentermine, cocaine and scopolamine, hydromorphone and morphine, psilocin and bufotenine, and LSD and LAMPA. And so for the results of this study, they found that spectral differences were observed for some pairs, but not for all. And so again, that really supported the use of DART for screening, but not necessarily for confirmation. And so just as an example of a pair where they did see spectral differences, for the methamphetamine and phentermine pairs at the 30 volts voltage for orifice 1, they noticed that each compound had the 150 protonated molecular ion and a 91 fragment, but meth fragmented and gave a prominent 119 versus phentermine, which gave a 133. So that was a pretty clear distinction between those 2 isomers. Conversely, something like LSD and LAMPA, even at the 90 volt orifice 1 voltage, where you're going to get increased fragmentation due to the collision-induced dissociation, the fragmentation was nearly identical. There's very few differences, and any differences that may have been present are not necessarily reproducible. So again, just further conclusion that this is a good screening technique, but maybe at least at this time not ready for confirmation. And then the third sort of study that they looked at was comparing the results with GCMS data, because of course, if we're trying to institute this as a screening technique, we want to make sure that it's actually giving us a valid idea of what is inside the sample. So they looked at 553 case specimens that had previously been analyzed and confirmed via GCMS, and they analyzed them using the DART TOF and compared to the results. Except for one specimen, in each case, the GCMS and DART TOF results were in agreement for the highest scheduled drug. The only specimen that gave a different result was a heroin sample that had an unusual cutting agent. The protonated molecular ion measured at 369.2147, which means that its carbon-13 isotope overlapped with a heroin peak. So I have an example. So you can kind of see that's really not a clear peak. It sort of has a big shoulder. Now, if you were looking at this DART data and you were maybe more experienced with DART, you can notice that 310 that's labeled as fluoxetine, it's a pretty common fragment for heroin. So if you were more familiar, you might sort of still be able to use this as an indication of heroin. But again, clearly it's just more justification that, especially in mixtures and without any sort of separation beforehand, really this has only been only a screening technique and not confirmatory.
So if you're looking at the timeline of how long it took us to actually implement this into casework, you can see that it took us a decent amount of time. We got the instrument in February of 2020 and then basically got shut down for COVID. We did have help from Ed Cisco to help validate it, which did move the process along a lot faster because we were really not familiar with this technology. And so we were able to run a whole bunch of the sections of the validation. And then in July, we realized that there was a high voltage leak that was causing the mass to charges to drift a little bit. So that was repaired, and then we had to repeat some of the validation because it did affect the accuracy of the instrument. So then we realized in November that we could use the software of the MS Axle that it had a sequencing tab, and we could actually create 1-minute sequences and run each sample that way, which is a lot more consistent with how you would run GCMS samples. So we decided to use that instead of these longer runs, and that did make the workflow a lot nicer, but we had to decide whether or not we wanted to continue scanning more voltages, or if we just wanted to simplify our process, use the 30-volt data, and get the benefit of some auto processing that the instrument would do. So we decided to use the auto processing and only scan for 30 volts since we were using it for screening. So we had to adjust our method of how we were going to sample a little bit. So the validation was completed at the end of the, December 2020, and then it took us a few months to write it up, but then it took another 5 months to get signed off for someone to use it in casework. And we're hoping by the end of 2023 that we will have everybody in the Pikesville and Hagerstown lab signed off to use it for casework. So it's still an ongoing process of training and competency to bring it online. Next slide. So the reproducibility section that we did, we ran a 15 component solution 5 times a day for 7 days. And this is an example of the 20-volt data. And you can see the dotted lines indicate the 5 MMU drift that was acceptable. And all of the data points are within that 5% tolerance. So it is very accurate, even with a complex mixture. Next slide. So for selectivity, we basically looked at different types of compounds and compounds that had other isomers, and we saw whether or not we would get scores that were higher for a compound that was not in the mix. So it worked as expected. We did see instances where another drug matched higher than the compound that we had, which is what we expect with compounds like methamphetamine that fragment pretty similarly. But it was able to differentiate between drugs like cocaine and scopolamine that are structurally not as similar. There were no instances where the compound of interest was not identified within the top 5 hits. And the search scores were above 750 for all of those compounds. So this was done before Ed's DIT software was the version that he has now. So we would like to go back and look at that data with Ed software and see how the scores benefit confirmation of the compound of interest, but that didn't exist at the time. Next slide. So we also looked at what solvent was best to use, and methanol was by far the best solvent to use. Hexane didn't do too bad, and acetone and chloroform did have some effects on the peak area, but there was still plenty of signal to be detected. And we did not see any solvent-induced drift or adduct formation with the solvents that we used, but that is possible with different solvents. Next slide.
Okay, so I talked about how we at DFS validated the method as a whole, but that was run on our instrument in the central laboratory. So once our regional laboratories got their own instruments, we basically performed an addendum to that validation where we verified that each instrument is still working appropriately and within the scope of that initial validation. And this verification process is actually listed in our operating procedures, which are available on our website publicly. So if anyone is interested, they can feel free to check that out. But basically, whenever we get a new DART instrument and after it's installed and the manufacturer is happy that it meets all of their required checks, we perform our own verification. And so the first thing that we'll do is we analyze our quality control check mix. And for us, that includes meth, cocaine, and nifazodone. So that basically gives us a range between the mass ranges that we're typically analyzing. So we'll analyze that 10 times within one data file, as well as 5 times in separate individual data files. And using the 30-volt data, we'll confirm that all of the protonated molecules are all measured within plus or minus 5 millidaltons. After that, we'll perform our own lower limit of detection study, again, on each of our individual instruments. And so this is sort of a compressed version of what was done for the entire method validation. So in this case, we'll only prepare solutions of 4 different drugs. So we'll look at cocaine, heroin, methamphetamine, and alprazolam. And we'll look at the same concentrations. They range from 0.01 to 0.5 milligrams per milliliter in methanol. We'll run each dilution 5 times and then determine the dilution level where the protonated molecule, again, strayed beyond that plus or minus 5 millidaltons. And for us, we had our instrument installed in 2015. And again, as I mentioned, it is the newer 4th generation instrument. And so I believe our heroin lower limit of detection was down to 0.03 milligrams per milliliter. And all of the other compounds were down to the 0.01 milligrams per milliliter. Next, we have to verify that our Mass Mountaineer is operating as we expect it to be. So we will load the applicable libraries, and then using the data that we generated from that lower limit of detection study, we'll search the 30-volt data against search lists, and we'll search the 90-volt spectra against spectral libraries, and just ensure that all of these searches will correctly identify the peaks of interest. We then will do our own selectivity check. So again, this is sort of a condensed version of the overall method validation. And so we'll use function switching and analyze 3 different isomer pairs. So methamphetamine, phentermine, hydromorphone and morphine, and cocaine and scopolamine. And we'll just ensure that those differences that we noted in the method validation can also be seen during our verification. And so obviously this verification is a condensed, like I said, version of the total validation. I think ours, we had it done maybe within a month, maybe 2. So it is done pretty quickly.
Okay, so as part of the NIJ project, we were trying to figure out if bringing the DART in as a screening tool actually helped us improve our workflow at all. So what we suggested was that we wanted to be able to screen with DART and then aim for a targeted confirmation. And the idea would also be that if the sample screened negative with DART, we wanted to stop at DART and not have to analyze it further onto GCMS. So we have not yet collected enough data to show that we can stop at DART with negative samples, but our plan is to collect data for 6 months and then look back to see if the DART would have potentially missed something without that GCMS screen. And then if there are CDS detected, we started developing these optimized methods to identify each class of compound. And our latest venture with the NIGA project is to try to create another general method that would detect most drugs and cutting agents, and would be just one method that the chemist would use. So we're kind of building a variety of options for confirmation, and we're trying to see if we can do a better job at identifying isomers or speed up the process. Next slide. So the way we implemented it into the workflow is that it takes about 10 minutes to warm the instrument up, get it ready to run the positive and negative control. For the beginning of the week, you run the PEG calibrant and make sure that the mass accuracy is good. And then you'll check it with an internal standard, which is our negative control, and the cocaine with internal standard, which is our positive control. We're using glass rods to dip into the sample to wave in front of the gas stream, and that's how we're introducing the sample. We are using the sequence chart to collect the data, and we are using Mass Mountaineer to search the data. And this is 30 volt data because we decided just to use the 30 volt. And then, so from start to finish to run a sample, it only takes a minute to run the sample and less than a minute to get your data off of the instrument. Next slide. So we did decide to implement an internal standard, and we did an internal standard study to see if there was any limitations in using an internal standard solution. We chose tetracaine because we don't see that in case work samples very frequently. And so there's a 0.1 milligram per milliliter tetracaine internal standard. That's what we use. And we found that it's helped us to make sure that our samples are within the range that it will get mass accuracy on the instrument. And it's also helped us to know if there is a sample that does not contain a drug, because you would just see the internal standard and you would not see any other substances in the search list. Next slide. So we wanted to see whether or not the internal standard would affect the ability to identify the common drugs that we see in samples. And we were able to show that there is a little bit of competitive ionization happening, but at the amounts that we have samples, it was not a problem for detection. We also wanted to know what concentration to set our internal standard at. And what we decided to do was to tie it in with our data integrity. So we previously had measured the background levels of drugs in our laboratory, and we had looked at what those levels would look like on the GCMS if we were to concentrate all the background that we had into one sample and ran it on the GCMS. And so we kind of wanted to do the same thing for DART, because we know it's a very sensitive instrument, and we were worried that we would be detecting things that were actually in the background and not in the samples. So we found that the limited detection for DART is a lot lower, or is higher than our background. But for GCMS, our limited detection is somewhere around 10 micrograms per mil. And when you use the internal standard at the concentration we set, the the area count of the internal standard is above 1,000,000. So when you threshold to 5% of that, you are not picking up the drugs that would be at a background level. So we did find that it would help us with data integrity, not have a result in our search list that was not from our sample. Next slide. And this just shows that all of the compounds were detected above 2.5% threshold of the internal standard. We chose 5% just to be a little bit more conservative, but you could definitely set it to 3% and still have good data integrity. One of the benefits to setting it a little bit lower is heroin does not ionize as well as fentanyl and some of the other things in a typical heroin mixture. So sometimes we'll see heroin, but it'll be below the 5%. And, you know, if you set it to 2 or 3%, you would be able to pick up heroin more frequently. Next slide.
Okay, so now I'll talk about how we use it in our workflow, because we do use it a little differently from Amber. But similarly, in every single data file, we'll have our calibration solution. So we use PEG 600, and the scanning range that we use is 66 to 600 Daltons. And so we'll use the PEG to calibrate the masses across that scanning range. And again, in every data file, we'll do our drug mixture QC check. So we don't have an internal standard that's in our samples, but we will analyze a drug mix with every data file just to make sure that the calibration is working appropriately. And as with before, we'll ensure that our measured masses are within plus or minus 5 millidaltons of the calculated masses. And we also run that QC check as part of our daily QA on the instrument. Now, our chosen sampling method, similar to Amber's, we use the glass rods that have been cleaned. And so we'll dissolve our samples into a suitable solvent, dip the closed end of the capillary tube in the liquid, and then hold that wand in the gas stream. And so we don't have the sort of automated processes that they're using. So this is done manually, and we can do in 1, 2, or 3 wanding events for each sample. And so this is sort of an example of what our chronogram might look like. So in this case, an analyst did 3 wandings of the PEG 600 mixture, 3 wandings of the drug mix, and then 2 wandings of a sample. And now frequently, most analysts will also do a wanding or 2 in between the drug mix and the sample of whatever solvent that they utilize. So again, since we're not using an internal standard sample, we don't have that. So whether it's methanol, chloroform, even water sometimes, they'll just wand their solvent just to show that, that there's nothing in that solvent that could be interfering. And where this actually falls in our analytical scheme, typically this will fall after a color test if that was appropriate for the particular case. So most of our analysts will use DART instead of where we previously were using thin layer chromatography. But again, this is still up to analyst discretion, and it's going to be up to whatever type of case it feels most appropriate. So if color tests indicated something maybe more straightforward like cocaine or meth, then typically most of our analysts will still do TLC before putting it on the mass spec. But if there's something more complicated or maybe inconclusive color tests or some heroin fentanyl mixture that, again, we just want something a little more selective than TLC, then we will typically use the DART for our next screening test. And just an example of how we might get some results for drug screening. So we have a mixture that has fentanyl, caffeine, and acetaminophen, and of course, DART-TOF will ionize all components. But as Amber kind of alluded to, especially with heroin, you can't necessarily determine the concentration of each compound based on their abundances. Depending on their proton affinities, that's going to sort of vary and can affect how much their abundance and ionization actually is seen. So as she mentioned, heroin really can very easily be outcompeted by fentanyl or by any other mixtures and any components in a mixture. So you can't necessarily determine the concentration, but you can potentially identify that there are multiple components in a mixture. And then something I really like the DART for in particular is mixtures that might have isomers present. So this is an example of maybe an illicit tablet where the 30-volt data indicated the protonated molecular ions for methamphetamine, caffeine, and utolone. But of course, utolone is a positional isomer of dibutylone and pentalone. So from this, we can't necessarily determine which isomer is present. And so you might go and say, well, let's go ahead and put it on the regular GCMS. But anyone who's familiar with these isomers will know that they are very similar mass spectra. They have very similar fragmentation. They all have the same base peak of 86, similar 121, 149 abundances. There might be some differences in the 41 to 71 range, but I'm not sure off the top of my head if those are consistent, and it obviously depends on where your mass scanning range begins. There may also be retention time differences, but another piece of the puzzle can, of course, be DART. And so since we do that function switching in every case sample, we can always then go back to our 60-volt data. And so this is just giving an example of kind of eliminating the non-isomer peaks in our 60-volt spectra. So we can see that the 138 and the 195 are likely coming from the caffeine, and we can see that the 91 and the 119 are coming from the meth. But then if we compare to the three cathinone isomers, we can see, and if kind of bear with me, obviously I know there's a lot of data on this slide, but you can see that pentalone and utolone both have a 188 versus dibutylone doesn't and has a 191 instead. Similarly, pentalone and utolone have a 218, which is not present in dibutylone. But conversely, dibutylone and utolone both have the 161 that's then not present in pentalone. And pentalone has the 205, which is also not present in either utolone or dibutylone. So just based on this 60-volt spectra, you can see that it's definitely much more likely that the sample contained utolone than any of the other isomers. So this, in combination with any retention time differences or the smaller GCMS mass spectra differences, can really help justify which isomer you might have. Now, I talked about how we're primarily using this as a screening tool, but something else that we have also been utilizing and have validated is using it for confirmation of pharmaceutical cases. And so where that comes into play in our analytical scheme is after we've conducted a visual identification and looked at the markings to determine what the manufacturer is indicating is present. We then have several options for structural elucidation to confirm that is actually what it is indicating is present. And DART-TOF is one of our options. So we can use GCMS, FTIR, or DART-TOF. And if those results are consistent with what the manufacturer is indicating based on the markings, then we can consider that a confirmation and go ahead and report. If it's not consistent, then obviously we have to go forward with a more full analysis. And so the reason we're able to do this is we're utilizing, again, that function switching of the orifice 1 voltage. Again, if you're interested, this was published in 2014 in Forensic Science International. And so what we're looking at here is we need to have at least one lower voltage, which we're considering to be 20 or 30 volts, as well as one higher voltage, 60 and 90. And that combination, and again, as long as it's consistent with the manufacturer's markings, are considered sufficient to confirm that pharmaceutical match. So our lower voltage, we're getting the protonated molecule, and our higher voltage, we're getting that fragmentation information. And just as an example, we have a 30 volt where we're seeing a 152 and a 300. And so when we search this just against our 30 volt search list, again, we can see the 152 is consistent with the acetaminophen, but then the 300 could be consistent with either codeine or hydrocodone. And so we move forward to either the 60 or the 90 volt data. And obviously there's going to be a lot of fragments in common. So in this case, both hydrocodone and acetaminophen and a codeine and acetaminophen preparation are going to have the 300, the 152, and the 110. But then in between those, we're going to see a lot of very reproducible and consistent differences. So for example, the codeine preparation generally has a very prominent 282, and also will have 243, 215, 225, versus the hydrocodone will typically not have that 282, and will also typically have a fairly prominent 199. So again, the combination of that protonated molecule with the fragmentation information, and again, assuming this is consistent with the markings, we're able to use that as confirmation. Now I'll let Amber go over a final consideration.
So as Jennifer was talking, I realized I should have mentioned that when we analyze our sample within that minute run, we do one dip of the internal standard, which acts as our negative control. And then we do 3 dips of the sample, and then we extract all of the sample together and the internal standard separately. And then those are what we put in our case file. We use the internal standard to our advantage to actually see if we have accuracy across the minute. So we're using that ion to drift compensate for that minute. And we're also using the software to auto centroid. So final considerations, the raw data from the DART is very large data files. So we actually had to have more data storage than we would have to just to back up a GCMS. So we have these external hard drives that store a few terabytes, and that can get expensive. The process data is smaller. So if you're storing process data in your LIMS system or something, it might not be like as overwhelming. But the raw data files can be a lot to back up. The additional workstation, you may want to consider it. It doesn't really take that long to run the samples now. If you had about 20 samples, you could run the samples in under 20 or under 25 minutes, and then you could do the data analysis for an additional 10 minutes. So that's not really taking up too much time. But if you wanted to have a second station where you could do the data analysis part so that you can not tie up your instrument computer, you can do that as well, because we only have one DART for about 10 chemists in the Pikesville lab. So any way you can free up the instrument, that would be good. The maintenance isn't too bad. We don't do too much maintenance, but you might need to open up the source to clean the source, and it's not too much more complicated than cleaning a GCMS source. You want to make sure you don't hit the wrong button though, because it will sound like a dying animal. Not that I've done. But you can also wipe down the orifice with methanol, and you can switch out a ceramic cap if the cap becomes dirty. We have a service contract, and the service contract is about $18,000 a year. So you get a PM with that. I would recommend it, but it's also not very cheap. And now we're
I'm going to add in, sorry, before we go to questions, I'm going to add into the maintenance. I believe this is all newer instruments, so I don't think it's just ours, but there is a isolation valve in between the ion guide region and the time of flight actual analyzer. And so that means that when we take down the ion guide region to clean it, we don't have to vent the entire instrument. So that time of flight is able to stay at the higher vacuum. And so that means that, that maintenance is really pretty quick. We probably get it done less than an afternoon, including, everything and retuning when we bring it back up. It's pretty quick. So I just wanted to include that. So that's really a nice feature.
Yes. You can go to the next slide.
Yep. So we're going to talk about some testimony tips, but before we kind of get into that, we wanted to know if there were any questions either on the validation, either at Virginia or the Maryland State Police, or how we incorporate it into our analytical schemes, because of course we both sort of do it with similar aspects, but then also very different aspects. So if anyone has any questions before we get into testimony.
I'll throw one question out real quick. Why did you guys choose to go with high-res mass spectrometry as opposed to low-res systems or MSMS systems?
Because you told us to. No, just kidding. Mostly because the higher res systems will differentiate between more compounds. So the lower res systems, without separation, you're not going to be able to differentiate between like compounds that are similar but not that similar in structure. So to get the more specificity, we went with a higher res one.
Yeah, I was going to say the same thing. Since there's not that separation, having that increased selectivity based on the accurate mass is really helpful. Also, just I think the ease of sampling was kind of the bigger driving factor than necessarily what comes in the tail end. I think that was sort of an added bonus, but I think when they originally were looking into it, that ambient was really kind of the big selling point.
I had a question come in. How many people were on each of your validation teams?
Oh boy.
For us, Ed and I did the majority of the validation until we got to the non-probative casework sampling. And then we had 4 chemists do that section. I wish we had a validation team, but it was just me and Ed.
Ours was before I was here, but there were only 2 authors on the publication, so I'm hoping they were the only 2 primarily involved. Obviously, we can check the acknowledgments for anyone else who may have been involved, but I think it was primarily those 2.
Sure.
Yeah, and the validation, we have like a package that you can find on NIST website, and that will have the validation plan, the validation summary, our SOP. So you don't have to start from scratch. You can, take ours and just modify it for yours. So I think that's helpful.
Another question was, what is the ballpark cost for the systems that you guys are using?
I think ours is $250,000.
Yeah, I was gonna say 220,000 just for the DART itself. Plus, you got to remember the 18,000 a year for the service contract, and that's not including all the stuff that went around it, like the electrical installation. So I think we budgeted over 300,000 to put a new one in a lab.
And obviously, that's going to depend heavily on the mass spectrometer you're using, but you guys are both using the same mass spec system.
Yes.
For pharmaceutical validation, with the expansion of designer isomers within compound groups, is there a point at which you can no longer distinguish certain isomers?
That is, I'm sure there are points at which you can no longer distinguish them. So again, that's kind of partly why the confirmation is only for pharmaceuticals. So again, you have to already show that it's consistent with that pharmaceutical marking. So we're not going to see bromezolam and confirm it just based on DART. Like, we're going to take that, a step further. There's some alprazolam isomer, it was like 7 amino or something like that. Don't quote me on that. That I know is sometimes a little concerning, but I'm not sure if we've had that in like a legitimate looking alprazolam preparation.
And then what does training look like for new chemists who are getting introduced to DART?
Can I take that one? Because I actually have a trainee right now. All of our training manual is online, first of all. So if you want to see the exact procedure, feel free to look that up. I can put the link in the chat. For all of our different instruments, we have a series of study questions they have to answer, as well as a written exam and some practical exercises. And for the DART in particular, the practical exercises are the training coordinator will give them at least 10 different samples, kind of ranging in different, complexity, and some might be a pharmaceutical confirmation, some might be representative of a screening, and they can't be run all on the same day. They have to be run separately. So you have practice, bringing the instrument up and, doing start to finish multiple times. The first kind of 2 or 3 are maybe supervised, and maybe kind of the training coordinators holding their hand a little bit more. And then as we get throughout the 10, the last, several are done completely independently.
Yeah, and just to add to that, like, one of the tips that I would recommend is seeing what buy-in you have for bringing new technology into the laboratory, because I think one of the reasons why our training has gone so slowly is because it was intimidating to a lot of people to bring on this new technology, and they didn't necessarily see the benefits of having this more discriminating screening process. So having that buy-in from the beginning will make this whole process a lot easier.
And then you both mentioned you sample solutions. Do you guys ever sample pure powders or pure pills in any way? Or is it all solution-based sampling?
Typically, it's solution-based. Every so often, if we have sort of a really weird matrix that we can easily handle with forceps or something, we can obviously put that right in the gas stream. One of the big benefits I really like to DART is, especially if I have like a really messy cough syrup or maybe something I'm kind of not sure what it is I can just dilute it with water. And if it's miscible with water, I can analyze that, which obviously I can't do on a GC. So that is kind of nice, because then once I have an idea of what it is, now I know how to proceed with what extraction. Or I know sometimes we've had chocolates, which we used to be primarily THC, and now we're seeing more psilocin in chocolates. So that's another thing where we could just, put it in methanol or just a little bit of the chocolate. I think most of us would extract the methanol, but you could, if you're really unsure what it is, just put that in there.
And we don't have as much flexibility built into our SOPs, so you have to use the internal standard solution. But in other applications, not for casework, but for a pilot project that we've done, we've analyzed swipes of baggies, and there's enough material on the outside of the drug packaging to identify what's in it. And there's a paper written about that called What's in the Bag. It's the primary author on that. So that has been pretty cool. But as far as casework, we would have to use that internal standard solution in methanol.
Cool. And I guess one last question before you guys continue on. Have you guys, either of you, been successful in integrating this type of data into your LIMS system?
We are not paperless yet, so no.
Yeah, we are also not paperless. So I do think the LIMS system would probably help with how to record the DART results on your worksheet. But at this time, we are not that sophisticated.
Cool. I guess you guys can go ahead and keep going to testimony, and then we'll take the rest of the questions at the end.
Okay.
Okay, that was good. So as with kind of any testimony, I think the most important thing is not to overcomplicate it. I'm just going to go back a little bit. I think it's really tempting when we have a new technology that we're excited about, that if someone were to ask, so what does this instrument do? And you kind of want to just tell them everything it's capable of. But I think it's important to remember to sort of take a step back. And so just kind of answer the question as it's asked. If it's asked, what this instrument does, at the end of the day, the answer is that it can give an indication of what might be present in a sample. At the less technical level, that's really all they want to know. So I think the best practice is to sort of get used to answering questions in maybe a more general way. We're really big on working on first pass, second pass, third pass answers. So again, kind of starting more general. And if they want more detail, they will definitely ask you for it. And at that point, that's when you can kind of get a little more technical. And I do want to share, we have the transcript of the first time that someone testified about DART in our state, which was conveniently the same person who did the validation. So I do have that transcript. I'm not necessarily saying this is the best, perfect, most amazing way to testify, but it is at least something that has been done and used and seemed to go well. So the question was just, what kind of machine do you use? And our analyst said, I used several. In this case, there's a new instrument that we use that was approved for use in 2008 called the TOF DART. He says what the DART stands for, and that is used for screening that I use in this particular case. So I think that's a really good example. Again, he didn't necessarily go into, oh, this is what the TOF does, this is how it works. He just said, I used it for screening. So there was some follow-up saying, okay, so you use the TOF DART in this particular case. He said, yes, it's a time of flight mass spectrometer. Now, he did go into a little bit of detail on that. I won't necessarily read it all for you, but he does sort of give a general overview. I like how he started with, the sample is dissolved in the solvent, which allows the material that's in the powder itself to go into the liquid. So he kind of gives a visual of how it's used rather than necessarily, again, starting from a more technical perspective. Obviously jurors like visualizing things, so you can kind of let them see, the small glass rod is dipped into the liquid. And so this is all sort of still, even though there's some technical terms, it's still kind of more of a layman's overview. And then at the end, it sort of says, analyzes those ions that come off of that, which are specific to the molecules that were present. There can be arguments about whether or not specific should have been used, but again, this is just an example. And I included this one because the attorney at this point got a little confused. Because he was thinking that the TOF DART and the spectrometer were two separate devices, rather than realizing the TOF part of that acronym is one instrument. So that was kind of a good opportunity for the chemist to take a step back and realizing, okay, that he's a little confused, so let me clarify. And so he does that. He says the TOF is the time of flight mass spectrometer, and then it's associated with the ionization technique of the direct analysis in real time. So obviously really important to make sure that you and the attorney are on the same page with their questions. And he does kind of go back and he says, there's actually one separate test, and with that, we've been allowed to use for screening purposes. And then after that, the answers are confirmed with a different technique. So again, just sort of taking it back to what's the important information, and that's just that we're using this for screening. And as they sort of continued with just, did that second test confirm or conflict? And it said it confirms them. And were there any inconsistencies? So again, that was just sort of the first time that we had testimony regarding DART TOF in our state. And even though he sort of started to go into the more technical side, it really didn't get above anything more than just ionization. At the end of the day, the really most important thing was this is a screening technique, and we then confirmed the results with another test. I don't know if Amber wants to talk about the analogies.
Sure. So at MSP, we love our analogies. So we have tried to come up with a perfect analogy for the whole process, and we don't really have one yet. But I'll give you the ones we are working on. To describe how the compounds can be measured by their time of flight, we use the golf ball, baseball, and basketball analogy, where if you use a bat and you hit a golf ball, a baseball, and a basketball with the same amount of force, the golf ball is going to travel the fastest to reach the end point. So, and the basketball would travel the slowest. So you would be able to tell which type of ball you had based on the amount of time that it took to travel. The horse race analogy, I thought it was genius, but my director didn't like my analogy because apparently I don't know that much about horse racing. But basically, when you have a bunch of horses lined up at a gate, you raise the gate and the horses leave the gate at the same time. And you would expect the horse that is the smallest and fastest to reach the end point before the horse that is the largest and the slowest. But apparently the size of the horse is not as accurate as a time of flight mass spectrometer. But the idea is basically that the gate opens, the compounds leave the gate, they travel a known path, and they reach a detector at a specific time. And the amount of time that it takes to travel that known path is the indication of what the molecular weight of that compound is. So those are the two analogies that we've been working on. We also have an analogy for collision-induced association. We think of it as aggressive bumper cars. So that would be when you're using 30-volt difference between orifice 1 and orifice 2, the compounds are not moving super quickly, so they can get from they can travel into the mass spectrometer without a lot of fragmentation. But as you speed up the travel of those compounds inside that chamber, you cause them to bump into each other more and produce more fragments. So that would be aggressive bumper cars, when the cars are moving really quickly and they're bumping into each other and losing pieces along the way. So those are the basic analogies that we have to explain the time of flight.
I love those.
They're a work in progress, but take what you need.
I mean, like I said, I think painting a picture for the jury is really the most important thing, and sometimes painting the actual picture is going to get way too complicated. So I'm a big fan of analogies too. I tried to think of some potential questions that are kind of more realistic to what I might actually be asked on the stand, and then try to think of how I might answer them. So we on our reports have to have, what methods we've utilized. So I'm coming at this question, kind of assuming I've already said, hey, I analyzed this sample using, this technique. And so they might say, okay, well, what information did you get from this test? And so again, just sort of keeping it very simple, I could just say something like this is used as a preliminary screening tool to give an indication of any substances present in the sample. Again, we're not kind of going into the details of the accurate mass. We're not going into the ionization, just kind of a very surface level, what information did I get from this? Now, if they press me on that, I can kind of go into that the instrument will determine the likely molecular formula, and I'm going to potentially assume that the jury might not know what the molecular formula is. So I could just follow that up with saying that molecular formula tells the number and type of each atom in the structure, which is going to be highly indicative of its identity. So again, you don't necessarily need to have a big chemistry background to kind of understand that, hey, this is not saying exactly what it is, but it's giving a very good idea. And if they really want to go more in depth, that's at which point where I'd probably throw in the analogy, because I'd say, you know, it uses the time of flight mass spectrometer, which means the compounds have in the sample travel a set distance. And then I'd probably use one of the Amber's analogies to just indicate that, you know, smaller molecules are going to travel more quickly. And so then we can use that to determine the molecular weight of those compounds. And now, if giving an example of using it for confirmation of pharmaceuticals, let's just say they said something to the effect of, how did I analyze item 1? I would say I compared the appearance of the tablet and its markings to an improved reference, which gave an indication as to its composition. I then used an instrumental technique to confirm that the contents were consistent with that initial assessment. And in probably 9 times out of 10, if not more, that's usually where that questioning would end, and we'd move forward to what was actually in that tablet. But if they want to know more about that instrumental technique and just say, you know, what instrument do you use, that's when I would go into what the name of the instrument is, as well as what it stands for. But usually on a first pass, I'm going to kind of try not to use the instrument names, just because sometimes words like mass spectrometer can be met with a little bit of a glazed look. So I'll try to say, again, just kind of very surface level what it was. And then if they ask, again, rather than going into the details of how the instrument works, at that point, I'll just sort of give what its name is. And then they might follow that up. Well, how did that confirm the presence of oxycodone? And then we kind of go back to what I said in the beginning with the screening, where it's going to determine the molecular formula. And I would also sort of say that there's a setting where higher energy is added to the sample, which can cause the molecules to break apart. And again, I don't necessarily need to go into what that's called or anything like that, but I can just say that, you know, that's reproducible, and the manner in which this occurs can lend additional support. And then the combination of that information with the pharmaceutical reference is sufficient for confirmation. Now, there obviously might be, if they, for example, did a subpoena ducis tecum and they have my case file, they might say, you know, well, your results for DART screening didn't indicate heroin, but your GC-MS analysis did, which, as we both mentioned, that can happen. So then we just sort of indicate that it's analyzing a mixture of drugs together rather than separating out each drug. And so some drugs like fentanyl get ionized very easily, whereas others like heroin do not. And again, this is just why this is a screening technique, and we move forward to a more selective process for confirmation. So just maybe some suggestions for testimony. Are there any final questions?
Awesome, thank you both. One question I had was, you guys talked about how you do your analysis and then how you testify to it. What goes into the report?
I don't know who wants to take it first.
You can go.
Okay. For us, we just have, you know, contents of one were analyzed and found to contain cocaine, and then we'll have a method statement, and it'll just say, you know, method, weight measurement, color test, DART TOF, GC-MS, and then we have underneath what those actually stand for. But that's really it.
Yeah, and then in our case file, we just use the tick, or the total ion chronogram, for the sample, and then we have the Mass Mountaineer printout for the internal standard, and then the sample. So it's 3 pieces of paper. We would probably use the DIT software because it's much easier to determine like what you have in the sample, because it gives you, a lot of times it tells you like what isotope peaks you have and that sort of thing. But it would print out, I think, is it still 3 pages, Ed?
Depends on how many compounds you have in your sample.
So we try to minimize like what we have to put in the case file, because we like to have smaller case files. But as far as reporting, since we're using it as a preliminary screen, it just has that we ran the sample with DART TOF on the methods used. And then in our notes, we would record what drugs we saw and what cutting agents.
Awesome. So anyone else have questions? You can either enter in the chat, enter in the Q&A, raise your hand and unmute. I guess another question I had was, have you compared the time of a DART analysis versus some of your other screening tools like color tests or TLC or whatever else you might use? And is there a time savings or not really?
We did do that for the EJ project. We measured like that. I mean, that was part of the process, was like measuring the old workflow and the new workflow. And I think like the amount of time it takes for you to do a color test is pretty close now to the amount of time it takes for you to run the DART. But the savings in time would be if you could do a targeted analysis and not have to run a longer method, or if, you know, with your targeted method, if you could not have to run as many standards for comparison. So our biggest time saving between the two workflows was not actually DART versus color test. It was the savings in the confirmation runs.
Yeah, absolutely. I was going to say the same thing. I think a lot depends on, in terms of time, how much you can batch together. So again, if someone has 20 cocaine samples, then it's probably going to be more time efficient to just run a TLC on one plate. But if you have a sample that has heroin, fluorofentanyl, xylazine, fentanyl, et cetera, rather than trying to get a TLC plate, which A, you're not really going to be able to differentiate all of those. And B, if you get a weird compound you haven't seen before, you're not going to know what TLC standard to run. So you're almost getting no information out of that. So then, like Amber said, you'd have to do a longer GC run. So yeah, I feel like the increased information is probably the biggest benefit.
It's also reviewable data compared to color tests,
Yeah,
so that's another benefit. Mm-hmm.
Good point.
For sure.
Question came in: how many analysts use the technique routinely in your laboratories?
Oh, that's a good question. We have about 10 chemists, and I would say probably 6 to 7 of them use it a lot. I think we only have 1 or 2 who kind of try to avoid it whenever possible. One of our requirements is we need to have the carbon-13 isotope for the molecular ion in the mass spectrum, which of course for some samples is not going to happen. And so if you don't have that, you have to use DART to get the molecular ion. So we have some chemists who really will only use it when they absolutely have to. But then we have others who have completely first said, I'm not doing TLC on another sample in my entire life. I will only ever run DART, and that's all there is to it. So yeah, and there's people in between those two extremes.
Yeah, our 2 newest chemists, the ones that are in training or just out of training, are the only ones excited to use DART. The others are being dragged along. But I think because our technology is relatively new, we only have I would say 3 chemists that would use it on a routine basis at this point. But I'm hoping by the end of 2024, more people are sold on the idea.
We had another question that was, we have to use a separation technique, and they consider DART a separation because the ions separate. What are your thoughts on considering this technique separatory?
I don't see how it's a separatory technique unless you have a QTOF, but I would say for the way we use it as a screening tool, you're getting all of the compounds ionizing at the same time. Maybe if you're using maybe a thermal desorber that you could ramp temperature wise, you might get a tiny bit of separation, but not enough, I would say, to call it a technique that's using separation.
Yeah, I would agree.
And then a practical question: how often do you need to replace helium tanks approximately?
We haven't had to replace ours too frequently, we've been able to get a few months out of ours, but we don't use it as frequently probably as Virginia.
Yeah, I'm not sure someone else orders the tanks, so I just always have helium when I need it. I'm spoiled, so I'm really not sure exactly how often.
Yeah, Virginia told us once a week when Bob Steiner was there, so I don't know if that's still the case, but I mean, it does use a lot of helium.
Yeah, I would say we at NIST at least, we are running 1 or 2 DART sources 4 hours a day, and once a week, once every other week is probably the rate at which we're going through helium. That is to say, there is a new part. I don't know if either of you are using jump shot, but there is a new approach with DART where you can pulse the gas and you can get severe reductions in helium consumption by using the pulsed setup instead of the constant stream. We don't particularly use that. I don't know if you guys do, but that is another way to reduce helium usage.
And we don't, but it's really cool.
Yeah, I mean, if you could use that setup, it would save you a lot of money.
Knowing what you know now, what advice would you give to a laboratory that's getting ready to perform a DART validation?
I'm going to mainly put that to Amber since I was not here for our validation.
I mean, we made a lot of mistakes during our validation that hopefully, if you were to look at our validation packet, you would get the benefit of us already having made those mistakes. I would say really just think about like, how do you want to implement it in your workflow? Because if you're really just using it for a screening test, 30-volt data is fine. And you get the benefit of some of the like, data processing on the end, which it just saves a few mouse clicks. It's not really that hard to do it, but like when you're analyzing like 20 samples at a time, that's not a bad thing. That's the main thing. I mean, I think the biggest decision that we would have to make if we were to implement one now would be like, how do we want the data like processed at the end? Do we want to use the DIT or the Mass Mountaineer? And there's benefits to both.
I also do want to, Amber had mentioned the benefit to an additional workstation, and we only recently got our additional workstation up and running. I say recently, it's probably 2 years ago. But it's a huge benefit because, it's only one instrument and ours is not as automated, so the person has to actively be there. But it's really nice that someone can be wanting their own samples and someone else can be at the other workstation going through their data. So that's definitely, if you can get that up from the start, that's probably ideal.
Mm-hmm.
What was your biggest lessons learned moment when implementing DART into your workflows?
I think I would just primarily the heroin not being as sensitive, and therefore, especially if you have strong fentanyl or quinine and you see that 310. So we don't have like a set threshold, like it's set at 5% when we're searching, but because we don't have any internal standard, we can lower that. So again, definitely if I'm looking at a 310, I'm probably going to lower that threshold and see if there's any heroin hiding there. Because there's definitely been times you'll see, heroin at maybe 1% of the fentanyl, and then you run it on the GC and they're 1-to-1. Maybe not quite that extreme, but sometimes it feels that way. So that's kind of my biggest pro tip.
Yeah, I think our biggest time saving was using the sequencing because the old way of doing it was not very popular. So the sequencing makes it a lot more like familiar to the chemist to walk up, program a sample, and walk away with their data in like under 2 minutes. I really like the internal standard, but I do see the benefit of like considering both options. I think we kind of try to standardize how people could do their data interpretation by using that internal standard. So we don't let them drop the threshold except to look for fragments. So we kind of standardized it, and there are probably going to be times where we wish we had a lower threshold.
Sure. And then have either one of you investigated a substitute for helium gas? Have you looked into nitrogen or argon or?
Yeah, we have not, but I know that Chip Cody's and others have shown that argon or nitrogen can be used. We've not personally looked into it.
Have you, Ed? I thought you looked at nitrogen or neon.
We have not looked at argon. We have looked at nitrogen. there's some caveats. You will see lower sensitivity with nitrogen as opposed to helium, but there are ways to boost that sensitivity. So I don't think it's been talked about yet, but my assumption is it'll be talked about next week. There's ways where you can put an interface on the front of your mass spec and actively pull sample towards your mass spectrometer, as opposed to the AccuTOFs that Amber and Jennifer are using, where there's no active pull. You're just relying on the mass spec to suck the sample in. When you have that active pull, you can actually enhance the sensitivity using nitrogen relative to not having that, and get sensitivity levels similar to those of helium. And then there also is some different fragmentation that can occur, or ionization and fragmentation that can occur when using the two different gases.
And that would be like also like using a nitrogen generator, not necessarily tanks.
You could also use the nitrogen generator in that instance. And as Sandra mentioned in the chat, you do see some odd electron radical formation when you use nitrogen for certain types of compounds. So relying solely on a protonated molecule isn't as reliable.
Yeah, and confusing is bad for us because we don't want to try to figure out. We don't want to figure out what happened.
And then let's see what other questions we have. Have either of you investigated negative mode in any of your analyses, or is it solely 100% positive mode and that's that?
We have a method for negative mode for GHB primarily, just because most of our analytes don't need it. I think I've used it maybe once, so I'm definitely not an expert in it. I have to revisit our SOPs every time. So we've dabbled in it, but not nearly as extensively as the positive mode.
Yeah, we validated the negative mode for GHB and the barbs, and so it works fine. It like you have to use it for GHB or you see GBL in positive mode. But the one thing we haven't really finalized is what to use as a positive control. So it would be a little hard to use it for casework at the moment because we have to just add that to the SOP. But other than that, it works great for GHB. But I mean, cannabinoids maybe. I don't know what else you would use it for.
So seeing no additional questions, we're going to go ahead and wrap up for the day. So we'll pull up again Jennifer's and Amber's contact information should you like it. We would like to thank both of them again for taking the time to present with us today. Their expertise and insight has been much appreciated. Again, feel free to reach out to them for any questions, access to resources, and things like that. Thank you, everybody, and I hope you have a great day.